Hello everyone, welcome to today's session. Our webinar this afternoon is on a slightly different topic than usual. We will be talking about ABO typing using molecular techniques, their importance and applications, followed by a live software demo of the newly released HLA Twin version 4.0. My name is Melista and I am the head of product at Omixon. With me today is my colleague, Libor, Kolesár, who is our senior field application scientist in EMEA, as well as a guest speaker, Professor Martin Písačka, the head of Transfusion department of IHBT in Prague. At the end of the presentation, there will be a live Q&A session where we will have the chance to answer any questions submitted during the webinar. Please feel free to type any of your questions at the question text box that appears on your screen anytime during the webinar. You don't need to wait until the end of the session. Finally, a recorded version of this webinar will be available within a few days from our website. So keep checking back if you would like to re-listen to this webinar at a later time. All registrants will receive a link by email once that is available. Finally, if you have any feedback that you would like to share with us, do not hesitate to send it to us using the sales@omixon.com or marketing@omixon.com email addresses. So I believe we are ready to begin. So as usual, I would like to start with a short agenda for today's webinar. We will be starting with an introduction of our company as well as ABO molecular typing kit. We will continue with our guest speakers presentation, which will be on ABO and other blood groups genotyping using molecular methods in the field of transfusion medicine. And then we will be following that with an HLA Twin demo for ABO genotyping, as well as the new features of HLA Twin version 4.0. And we will be ending with a questions and answers session, as mentioned. So to start with, a little bit of an introduction. So first of all, a bit of a company introduction for the people that are not as familiar with Omixon. We are a global molecular diagnostics company with headquarters in Budapest, where also our software R&D and assay R&D and other operations are located, as well as some offices in Cambridge, Massachusetts, where the US operations are based out of. Our existing products currently are the Holotype HLA, which is a pre-transplant assay and software product, the Monotype HLA kit, which is used for disease associations, the ABO kit, which is our focus for today, which is a molecular blood group typing kit, HLA Twin, a software focused for clinical purposes, as well as HLA Explorer, which is a software that is focused for research, mostly. In addition to all of these existing products, I wanted to highlight a wider ecosystem that we have built to support our customers and all of the clinical laboratories in the field of transplantation. This includes liquid handling automation, epitope analysis for HLA, using the Purge algorithms, as well as integration of our software with various LIMS systems. If you have any questions or would like any information about any of these ancillary ecosystem material, let's say, feel free to contact us anytime. So let's focus a little bit more on the monotype ABO, which is actually the ABO molecular typing is our focus for today. The ABO kit that Omixon offers contains ABO-specific primers that amplify a particular gene, library preparation reagents, indices, pre-plated indices, as well as analysis software, which is done through HLA Twin. The kits exist in two configurations for analysis of 24 samples or 96 samples. There are multiple index plates of multiple configurations that allow one to run up to 288 samples per run. And the kit has been available since Q4 of 2018, so for about a year now. The workflow, or the kit rather, It uses pre-mixed master mixes for amplifications. So you just need to add your ABO specific primers and DNA, you're ready to go. The kit uses the same workflow as holotype HLA version 3, for those of you that are familiar with the workflow, and I will mention it briefly in the next slide, which is very convenient because it does require less training and QC for the laboratory. The primers amplify from intron 2 to the 3' UTR, resulting in a 7 kb PCR product. And it does offer allele level genotyping results. So as you can see in this picture, you get a result with the phenotype displayed at first and the allele level genotype following after that. One of the best advantages and biggest advantages rather is the ability to combine the ABO and HLA libraries in the same run, which makes things easier and cost effective in the long run. So speaking of the workflow, I wanted to present this picture so I can break things down a little bit. The workflow requires about 3.5 hours hands-on time total from beginning to end, and it does offer a result from the DNA sample to answer in less than 48 hours total time. So as you can see in this particular picture, the process can be split into 4 different portions. We have the sample preparation portion, which would be the ABO specific amplification, followed by library preparation, continuing into sequencing, into an Illumina specific sequencer, and finally the data analysis using HLA Twin. And on this note, I would like to give the floor to my colleague Libor, who will be presenting our guest speaker and moving forward.
Thank you, Effie. And now let me welcome you today, Dr. Martin Písačka from Prague in the Czech Republic. He is a very well-known eminent Czech immunologist and transfusiologist. He is currently head of the Department of Immunohematology at the Institute of Hematology and Blood Transfusion in Prague. And he is also head of the transfusion section in the same institute. He is a member of the Czech Society of Transfusion Medicine and also of International Society for Blood Transfusion. He is also very active in other roles, like supervisor of the company providing the external preparations and testing for transfusion laboratories, or associated editor of the scientific paper called Transfusion Today. Under his supervision, the Department of Immunohematology became a national reference laboratory for the whole Czech and Slovak Republic. His laboratory is providing a full spectrum of tests for blood donation or routine assessment of the patients before the transfusion, especially in case of patients after the stem cell transplantation, where an early detection of changing the blood group from patient type to donor type is crucial to assure a correct selection of the transfusion component. His laboratory also provides services for the Children's Hematological Department at the University Hospital. And as a reference lab, they perform the most complicated immunohematological tests for resolving difficult cases of antibodies and antigens requested by the whole Czech and Slovak Republic. So, Martin, the floor is now yours.
Hello everyone. I would like to present about ABO and as well blood genotyping using molecular methods in the field of transfusion medicine. Immunohematology, as you might know, is a blood group science which has begun in the beginning of 20th century in Vienna when Landsteiner discovered ABO system. The whole century after this discovery could be called century of serology, when a massive increase of new techniques made possible to detect more and more antigens and antibodies, and the knowledge increased to nowadays a big amount of data of blood groups, antigens and systems. At the end of the century 20 and beginning of 21st century, the cooperation of classical serology with molecular biology techniques began, and now genotyping for blood group alleles represents important tool for solving complicated immunohematological cases. How the situation is complicated now could be illustrated that we know now 360 blood group antigens, and even more than 700 when we include variants and partial antigens. These antigens are grouped into 36 known blood group systems, and in this year a new blood group system, Augustine, was established, and 13 new antigens was also recognized by the terminology committee of IHBT. Others are grouped into 5 collections and series of low frequency and high frequency antigens. What are we speaking about? The system is a group of related phenotypes, and the relationship is based on antigen antibody reactions with defined and genetic characteristics. And background of this phenotype is genetic information, genotype. Blood group genes, we have now 47 genes, so more than systems, because some systems have more than one gene, and 1,700 blood group alleles, and known more than 60,000 of base pair of coding DNA for blood group genes. Information about blood group systems and blood group antigens are available in several databases. One of them is the IHBT database, available on IHBT web page. This database is based on SNPs, so nucleotide polymorphisms or exon rearrangements. Then there is a larger database containing larger sequences, but there is a problem with public availability of the database, is this blood group antigen gene mutation database. And finally is a very good database for RH system called RhesusBase, with detailed information about RH, D, and CE gene and alleles. Blood group and different alleles arise from point mutations or insertion deletions or gene rearrangements, crossing over gene core measures, and resulting proteins which carry antigen characteristics. We can recognize 2 big families of blood groups, 2 different families. One is biochemistry based on carbohydrate chains, and this group contains the most important blood group system, the system ABO, which is the most important system for transfusion, and is also linked to transplantation medicine. The second group is the protein group. We have proteins which act as receptors, adhesion molecules, membrane transporters, membrane enzymes, and proteins associated with complement regulation. The difference of these 2 groups is in the expression of genetic information. The first group, the carbohydrate groups, the genetic information is coding for enzyme glycosyltransferase, and the antigen is the product of this enzyme which transfer the sugar on the terminal position of the saccharide chain. The second protein group is different in the sense of direct expression of gene into protein structure, and the differences in amino acids or in the protein structure are blood group antigen of the blood group systems based on protein or glycoproteins. ABO blood group systems is a little bit complicated because we can distinguish ABO antigens on blood cells, but also on tissues. ABO system is so-called histo blood group systems. On blood cells, on red cells, ABO system antigens are expressed by red cells themselves and came from mesodermal tissue, and the backbone is the chain type 2 chain in contrast to type 1 chain, which is controlled by SE and LE genes, which is coding for ABO backbone, or the backbone for ABO antigens on epithelials. These terminal sugars occur on the terminal position of the oligosaccharide chain by action of glycosyltransferases, which can use two different sugars, either N-acetylgalactosamine in case of A1 and A2 transferase, and D-galactose in case of B transferase. In case of blood group O, we also have genes coding for transferases, but these genes are coding for non-functional proteins. So the chain remains uncovered with terminal sugars. It's the backbone H antigen is present, and no A, no B antigens could be detected on red cells or on epithelial. On this slide, rather complicated, you can see the way of creation A and B, or non-A, non-B, so O phenotype by action of glycosyltransferases A or B. And also on the lower part of the slide, you can see the backbone for Bombay phenotype when non-active genes regulating the H antigens are present. So those written with lower cases SE and H are coding for the Bombay phenotype. Here you can see the database, and you can see the first page and the last page of the database for ABO alleles, and you can see that the last position is 323. So in the field of ABO alleles, we have multiple alleles, but in contrast to that, we have only the two sugars, A sugar, the N-acetylgalactosamine, and B sugar, D-galactose, which are transferred in different quantities and on slightly different backbones in different A and B phenotypes. The ABO expression is also present not only on red cells, but also in secretions, and those presence in secretion came from epithelial tissue. So we can have ABO antigens on red cells and on epithelials in secretors who have active genes H and genes big SE. Then we have non-secretors who have ABO antigens only on red cells and not in epithelials and in secretions. And then rare groups where there could be discrepancies between phenotype and genotype, para-Bombay and Bombay, where in para-Bombay antigens of ABO systems are absent on red cells, but are present in secretions and on epithelials. And the true Bombay, there is absence of ABO both on red cells and epithelials. Molecular genotyping is a principle based on nucleotide complementarity and the PCR reaction. And the final genotype is then analyzed by different methods like PCR, SSP, RFLP, real-time microarrays, and different sequencing techniques. Why we need genotyping in transfusion medicine when we have a lot of serological methods? It's necessary because serological methods have many limitations. One of them is in multi-transfused recipients, when we need to differentiate own and transfused red blood cells phenotype, which is important for antibody identification, for prevention of alloimmunization, and for selection of best matched transfusions for recipients of multiple transfusions, especially for sickle cell disease patients. Other category of patients which is beneficial from genotyping are patients with autoimmune hemolytic anemia, where the red cells are coated with autoantibodies, which are disabling serological blood group typing. And in this case, genotyping can solve the problem and help to find optimal transfusion therapy. Other field of application of genotyping methods is solution of unclear, weak and partial and variant antigen typing results, both for the main important antigen systems, ABO and RHD. In case of RHD, the knowledge of weak phenotype helps us to distinguish between weak D and partial D. In recipients, then weak D can receive normal D positive transfusion, while partial D need D negative transfusions. And the knowledge is also important for pregnant women, where weak D phenotype pregnant women don't need to receive anti-D prophylaxis, while partial D need this application. And furthermore, we can also look for a fetal genotype from maternal plasma by non-invasive techniques. We can detect fetal DNA in maternal plasma, and we can indicate the need of prophylaxis in the mothers carrying D negative mothers carrying D positive child. And there are other applications of genotyping methods when we are looking for antigens where serologic reagents are limited or unavailable, like Tombrock, Colton, Bell, and so on. And also we use genotyping as a confirmatory method for before inclusion the donor into rare donor register, so we can exclude the presence of alleles coding for weak antigens like DAFI-X, or we can confirm the true negativity for high frequency like Celano, Bell, and others. Important knowledge is also exclusion of null alleles in donors used for preparation of diagnostic red blood cells, because for screening red blood cells, it's necessary to have strong expression, so homozygous expression. So we need to exclude false homozygous which can have lower antigen density than through homozygous. For example, in DAFI-A, where the DAFI-null or DAFI-X can complicate, can lower the antigenic amount on red cells membrane. Which genotyping is available in these days. So we have several genotyping systems, PCR, SSP with gel electrophoresis, which is a classical one, and the new systems are blood chip using Luminex platform or Fluogene, which is using fluid detection by immunofluorescence, and bioarray bead chip. I will show you shortly each of this platform. Classical gel electrophoresis are either kits from the BA or Inotrain and are available for detection of alleles, basic alleles for ABO systems, RH system, Kelky-Duffy, MNS, and rare screen. These methods are traditional, easy to instrumentate, but depends on several factors like quality of polymerases, and there's problem with toxicity of ethidium bromide, and sometimes subjective evaluation and not automated. Here you can see result of ABO, PCR, SSP, and this detection is limited only on detection of alleles A2, B, and O1 and O2, and A1 detection is per exclusion. So if there is no other allele, the genotyping is concluded to be A1. But as we have shown on previous slide, in ABO database, there is more than 300 different alleles. So you see that this routine technique is limited only to basic alleles. Gel electrophoresis of partial D, you can see on this picture. The most important partial D, D6. Type 1 is the upper one when exon 4, exon 5 is replaced in the gene with exons from CE gene, and type 2 has replacement of exon 4, 5, and 6, and corresponding gel are lacking bands in positions of corresponding to these exons. Other technique is blood chip ID from Grifols Progenica, based of multiple analyte profiling, when fluorescence color microparticles are covered with nucleotide probes, and evaluation is based on flow cytometry principle, and we are able to detect erythrocytes alleles and basic erythrocytes alleles, and also platelets alleles. Principle is that hybridization in presence of specific alleles is completed in case of complementary sequence, and then fluorescence is detected in case of positive match. Fluogene system is using endpoint fluorescence detection modified Dagman system, and it's quite rapid and easy to work with system. This system can detect RH exons and RH variants, Kelky-Duffy, weak Ds, and when we are speaking about ABO genotyping, you can see that the spectrum is limited to basic ABO genotypes A, A2, B, and O1 and O2 alleles. Then there are rare kits and KKD and MS. Then bioarray bead chip is based on elongation multiplex analysis of polymorphism, using also colored beads with nucleotide probes, and the reading is in this test by fluorescent microscope and evaluation by specific software. V-chip is able to detect the variants of RH, KIT, Duffy, and other blood group systems, and also variants of CE kits, which is especially important for African population, and also RHD, most frequent D types. But in this kit, ABO typing is not possible. Current methods which are available are limited, and we can be facing discrepant results between phenotype and allelic-oriented genotyping in case when new or rare single nuclear polymorphisms are present, when such probes cannot be included in routine kits because there are limited number of probes which could be used. Also, limitations is in women with DEL genotype because they have that DEL gene, and in such situation we cannot test for fetal DNA. And also we are limited when expression of specific gene is regulated by other genes, like in Lucerne null phenotype in Lu type, also in Bombay phenotype in ABO or Duffinol phenotype, where the GATA mutation is present. And also we can have atypical results and complicated interpretation in case of chimeric individuals. When solving discrepancy of current genotyping method, we are using sequencing. First is the classical sample sequencing, and now the next generation sequencing is coming, and it will be big help for solving complicated cases and discrepant cases. And in this case, for ABO, this solving of the discrepancies is critical for transfusion and also for transplantation, because ABO is histo blood group systems. These discrepancies could be either inherited, those rare alleles which were listed in the DBRBC, and the ABO system is typical that it's simple phenotypes, only sugars specific for A, and acetylgalactosamine, B, D-galactose, and no sugar in case of blood group O. But we have numerous alleles, more than 80 alleles for non-functioning transferases in O phenotype, 30 alleles for A, 15 alleles for B, and dozens alleles for weak subtypes. Furthermore, ABO expression is regulated not only by the ABO gene, but also by the effect of genes regulating the precursor chain, gene for big H, little h, and SE, big SE, little SE alleles. And we can have also discrepancies, altered expression of ABO antigen in dysplastic hematopoiesis, in hematological malignancies and dysplasia, and also can be facing different results after ABO incompatible bone marrow transplantation. In such situations, all genotyping results should be interpreted together with serology results before issuing the recommendation for compatible transfusion or transplant. And next generation sequencing is now coming to help us, and we are in the beginning of working with NGS commercial kit from Omixon, which is using Illumina platform and is examining ABO gene in exons from exon 2 to exon 7. So we are now in the phase of the first tests, and we will be happy to present our results on other transfusion congresses. Finally, I will show you some interesting cases from our laboratory, which were presented on IHBT Congress in Basel. These are new partial Ds found in last years in our laboratory, which are based on one amino acid exchange, which are quite critical for antigen expression. What is seen on the upper part is some epitopes are missing due to single nucleotide exchange in the RHD protein chain. And finally, one strange and interesting case of healthy blood donor who was found to be chimeric and has both population of A and O genes. And in this person, we have found more sets of alleles that it's normal. So we can see almost a double set of alleles from father and mother. This is represented in flow cytometry for red blood cells, and also on DNA polymorphisms that maternal and paternal alleles are present in higher numbers than in a normal individual. This finding was interesting because the person is a healthy man donating blood 70 times, so it's no problem for his health. With this good message, I am ending my presentation, and ended with a picture of blood of river Vltava in southern Bohemia, and wish you and wish us also interesting findings with NGS typing for ABO. Thank you for your attention.
Thank you very much, Dr. Písačka. And now I would like to give the audience the introduction of the new features of HLA Twin version 4. So in the meantime, you can think of any questions related either to the presentation or to the product or to the software, and we will address them later on after the software presentation. So at first sight, nothing really dramatic has been changed on the first page, because the biggest changes actually appeared inside the software itself. So the first change is, for example, that we have increased the response, we have decreased the responsivity time of the software. So when you are opening the folder with the results and data, it will open immediately. And second, which cannot be actually demonstrated now because it needs the connection to the Illumina instrument, is that the users of Miniseq and also the users of MySeq who have installed the local run manager can also use the automated data analysis. The settings of the automated data analysis now became even easier. So in order to do that, just go to application settings, and under the configure automatic analysis, you just need to set up the path of the folder where you store the FASTQ data, and that's it. So let's come back to the genotyping dashboard, the home screen. And you see the first change is down at the bottom, because you can see here the info pane, which will contain all the information about the result files or the FASTQ files, which database has been used, which software has been used for the analysis of that result or of that sample. Another feature, new features will appear farther on the results screen. So if we go to check the results, we will immediately see a big change. The experienced users know how this picture looked like before. Now we have changed the display of the results a little bit to the more table format. Some additional informations have been highlighted. For example, if you have a low expression allele, if you have other ambiguous alleles expected there, or if there is a message that the allele is not expected, for example, like in this case of DRB3, because there is no DRB3 allele expected if you have a DRB104 positive sample. Further on, previously we have the traffic lights related to the QC metric presented at each result, at each level, at each allele. Now, because it is QC related to the whole locus, we have moved the QC traffic light to the top. And next to that, there is another flag describing whether the result is heterozygote, like in this case of HLA-B locus, or if the result is homozygote, like in this case of DQB1 locus. Next, here you can see the status of the result. By default, it is a gray tick mark, expressing that this is a unprocessed sample. When we go farther in the approval process, the tick mark will gray into orange, meaning that this sample is ready for the final approval, and then at the end it turns to green, meaning that this result has been approved. The time symbol next to that is a button that will open the history assignment of the specific sample. And final upgrade of this screen is here, the collapse and expand button. It's nice that you can see a lot of details about your sample, a lot of results, but if you have more than one sample, you will not be able to see a lot of results on your screen. So it's convenient to be able to shrink actually the content of the table, like I do now. So you will see that each sample will now occupy only one lane. You will still see the QC metrics and homozygosity, heterozygosity, and you will see only one or two alleles. And now you can realize that some of the numbers are actually turned red, which means that at the fourth field position, in case of HLA-A locus here, there are more results, there are more alleles than only O2, O1, O1, O1. And now let me show you how to analyze the ABO genes, because we were talking about ABO the whole webinar. So I will show you how to analyze the data. So what you need to do is just open the folder where you store your ABO data from the Lumina sequencer. Select the FASTQ data, click on Advanced Genotyping button, and go to Gene Targeting tab. Here you have an option to select the gene family of the genes you want to analyze. So in our case, we will just click on the ABO, and we will click on Finish button, and the analysis will start. If you do the ABO typing routinely, you can even create a typing protocol, which will appear here in the beginning, and you just select the protocol for the ABO, and you click on Finish, and it will trigger the analysis. And here comes another new feature of HLA Twin v4. This is called Typer Scheduler, which is accessible through this button at the top. So I will now open that. So you see that now we have launched the analysis of 3 samples. First is now being analyzed and 2 are still waiting for the completion. And here is a new feature. So if you, for example, need to prioritize the last sample to be analyzed first, you can select it and you can simply move it. And then once this analysis is finished, it will start immediately analyzing the other sample. In Typer Scheduler, you can remove the sample from the queue if you just made a mistake and you don't want to analyze it, or you can just simply stop the current procedure if you don't need to analyze that sample. So that was actually everything what I wanted to share with you. New HLA Twin version 4 features. And now let me give floor back to Effie, who will continue with the webinar.
Thank you, Libor, and thank you, Professor, for your presentations. This was definitely very useful and very nice to hear. We will have another session. We will have our session of question and answer. However, before we do that, I would like to let everybody know that we will be making some product announcements at ASHI on September 26th in Pittsburgh. Anybody who will be attending ASHI, make sure to come join us for the announcements and also to see what we have been working on. And if not, make sure you follow Omixon's website and social media for the updates, and you will know everything that is going on. And on this note, we are ready to take questions. So let me just look through the list of questions here. There is a question about the availability of the ABO monotype kit. As I mentioned earlier, that is already available. So if you have any questions, feel free to contact us and we will be happy to make arrangements to send you one.
I can see one question coming from our distributor. It's actually related to your presentation, Dr. Písačka. So there is a question if all blood donors are genotyped, and if not, what are the criteria for the selection of their molecular-based genotyping?
It depends on which country are we speaking about and about which genotyping is done. For example, in Switzerland, all blood donors are genotyped for RHD. So even the weakest form of RHD, like DEL, could be detected, and immunization of patients with this weak alleles is excluded. In our country, we do not type for this DEL phenotype, and we genotype donors and patients if we have some finding of discrepant or weak antigens. And also we genotype when confirming rare phenotype for blood donors who should be included into rare donor registry.
Okay.
I see another question here. Are there any other NGS systems for genotyping of other erythrocyte antigen systems right now?
For my knowledge, I know that there are some in-house NGS techniques used, for example, for RH genotyping. Some results were presented on IHBT Congress in Basel this year. but not commercial system.
That will be very interesting to see soon. Yeah.
I can see some technical question. Is it possible to combine the HLA and ABO libraries together? Which is a very, very smart question, because if you have a capacity of the sequencer big enough, then why not to combine the HLA and ABO libraries? Now, well, do you want to answer?
Sure, yes. So if I didn't mention it already, it's my omission. It's definitely a possibility. A lot of laboratories do this in routine. So a laboratory that runs their HLA typing using NGS with a holotype kit, for example, they often do add several samples in the run for ABO genotyping as well. It just streamlines things in the workflows in a very easy way, and it ends up making things very cost effective as well in the end. So yes, this is definitely possible.
And it's a good advantage for laboratories who combine both needs of genotyping for patient and for transplantation and for blood donors. It's
absolutely. Yes, this is absolutely. I think these are all of the questions that we have right now. I don't see anything else. If anything else comes to mind, however, we will be happy to answer any questions you may have, or if you would like more information, feel free to email any one of us or support@omixon.com anytime. And we are looking forward to seeing you in our next webinar in the next couple of months. Thank you very much, everybody, and have a great day.
Thank you.
Thank you.